Hey everyone, welcome to Biochem. In this video, we will define the cell. Okay, let's start with a simple definition. The cell is the basic structural, functional, and biological units of all living organisms. Everything that is truly alive, from a tiny bacterium to a massive whale, is made up of those units. All cells fall into two major categories: prokaryotic and eukaryotic. Prokaryotic cells are simpler, and eukaryotic cells are complex. Prokaryotic cell has no nucleus, no membrane organelles, whereas eukaryotic cell has nucleus, has membrane organelles. Example of eukaryotic cell is animal cell, plant cell, which is multicellular, and an example of prokaryotic cell is bacterial cell, which is unicellular. Cells are tiny, often invisible to the naked eyes. To study the fascinating internal structure of the cell, we need to use microscope. There are two primary types we need to know for cell biology: light microscope and electron microscope. Before comparing the light microscope and electron microscope, we need to know what is magnification. Magnification is how many times larger the image appears compared to the actual size of the object. And resolution is the ability to distinguish between two separate objects that are very close together. Moving into light microscope. Light microscope use a beam of light, can view live or dead specimen. It has a maximum magnification 1500 times and a maximum resolution of 200 nanometer. It can form colorful image. On the other hand, electron microscope use a beam of electrons. Specimen must be viewed in a vacuum. Electron microscope can view only dead specimen. It has a maximum magnification of 500,000 and a maximum resolution is 0.2 nanometer, which is higher than light microscope. And it can only form black and white image. Always remember that light microscope is for observing living process and overall cell shape. Using light microscope, we can see cell and the organelle. And electron microscope provide a detailed, high resolution internal structure. Using electron microscope, we can see inside the organelle. For example, the detailed structure of mitochondria, ribosome, protein. Thank you so much for watching this video.